right. Good evening, everyone, and welcome to the Talking 3R Science webinar today. We're very happy to have a very interesting guest today, and I'm gonna hand over directly to Georgia, who's gonna introduce David and also the topic of today.
Thank you, Peter, and welcome also from my side, everybody. Today we are really the honor to have a very important speaker. It's Dr. David Pamier. He's from the Lausanne University, where he's a postdoc researcher. David has traveled around for his work. So we met at the Joint Research Center in Ispra in Italy. He also did his postdoc at the Johns Hopkins University at the CAT Center. And now, as I said, he's in Lausanne. But today we will not speak about his research that is more on neurotoxicity in vitro assays. It's about a good cell culture practice that is very important for very different fields, and also is evolving with evolving of the technologies. So yeah, I'm very happy, David, to have you here. And while you set up the presentation, I would like to ask already our audience to reply to the first poll that we normally do at the beginning of the webinar, is to understand who you are and which is your fields. Yeah, so we have quite a number of people from biomedical science, second from toxicology, together with other. If you want, you can write in the chat what is other. And then we have also some participants with the regulatory science background.
Okay, thank you everyone that wanted to join us, and also for the invitation to be here talking about GCCP. GCCP is kind of a tough topic, I think, for many people, and sometimes, you know, I feel like we are like screaming to the ear of some people, like, you have to do GCCP, it's really important, and people are like, can you please leave me alone, right? So today, because we have a broad audience, I thought that instead of, you know, going into really in detail on the guidance, so people might fall asleep, I think I'm gonna change a little bit the strategy, and I'm gonna try to convince you what are the problems that we are facing at the moment in cell cultures, and then some examples of bad practice and why good practice is important. And then by the end of the practice, I will show you some examples of how these guidelines can help us to improve our cell cultures. Obviously, the work that I'm going to present here is not done only by myself. There were many authors, and I don't want to get credit of all this work. So just take into consideration that I'm just here representing a huge group of people working on this topic. So as Gerhard already described a long time ago in 1999, these guidelines, the GCCP guidelines, are trying to establish minimum standards for cell cultures and tissue cultures in order to facilitate the interlaboratory compatibility. So basically, these guidelines are based on 6 principles. The first principle is to try to understand our model and cell cultures, but also the factors that will affect the cell culture. So for example, the media that you are using, or what type of cells you have. The second principle is based on basically how to maintain the quality and assure the quality of the model that you're using, the cell, how to maintain integrity, the validity, what type of controls or endpoints you need to measure to make sure that every time you perform an experiment, it works well. Then the third principle is related with reporting and documentation, and I will give you some examples later why this is so important. The principle 4 is based more in the protection of the user. So what type of things or measure we need to do to protect the individuals working in the cell culture lab. Then the principle 5 is related with the laws, legislation, ethical principles, and what type of rules you need to know, or laws you need to know to enable to perform the practice in a proper way. And the last one is related with the training. So how we can train the people working in the lab so they can do a good quality. Right. So there were 2 main documents, and I will talk them a little bit more in the end of the talk. But in case you want to have a look at these documents, there's one in 2005, and then there's an update that was recently published in 2021. So the idea of this cultural practice, right, is to so basically give you some tricks or what things you have to control in able to ensure reproducibility, reliability, credibility, acceptance on their assays. And why is this so important? And I'm not sure how where you are, but in the last years, we have had this boom of reproducibility crisis, where many scientists has been raised this concern that many of the science that we are producing actually are not, we are not be able to reproduce that. And this became really famous when Nature published in 2015 this survey, basically, where they collect information from many scientists, and they found that more than 70% of the researchers have try and fail to reproduce another scientific experiments, and half even failed to reproduce their own experiments. So that means that we are doing something wrong. And there are big discussion about if it's a crisis or if it's not a crisis, but obviously it's a problem that we need to find a solution. And this is, has been coming after that. Many other things have been raised, especially in cancer research. There has been many attempts to take, for example, there was an example where they tried to get the most influential preclinical cancer research papers and tried to reproduce them. And they found out that they were able to reproduce only 11% of this research. And this can go for different studies. They found also that for example, from the National Institute of Health, they found that 40% of these oncology studies that were funded by NIH were really ambiguous and unclear, and they were not also able to reproduce. And this is not only important in terms of scientists. In science, obviously, we are losing the quality of the science, and scientists normally is based on science. So if it's not possible to reproduce, we have an important problem. But also we have to take into consideration the amount of money that we are basically throwing to the bin. And the PLAS Biology published an article in 2015 when they thought they calculate that probably the reproducibility clinical and preclinical research have cost around $28 billion. So you can imagine how much money we are losing because of this irreproducibility and in some extent bad practice. But why is this happening, right? So there is several options, there are several factors, and obviously good cell practice is one of the factors, but we have other factors that we need to comment, like for example, the pressure to publish in science, right? We always need to publish, we need to be the best, and then sometimes we have to hurry to publish the first, and then we don't take into account the quality controls or the quality that we need to actually perform to produce good science. And also sometimes there's we have a negative results, we don't want to publish them because, okay, maybe if we publish these negative results, we are going to have some problems with the reviewers. Also, there is a lack of understanding of fundamental statistic principles. And it was also an article in PLAS Medicine talking about the statistic analysis. And they found that basically the 50%, that you have 50% chance that the results that you are presenting in the paper are not true. There is also insufficient mentoring from some senior scientists, especially in big groups where you have 20 people and they don't have the time to teach everyone. Hyper competition, there is a big problem in poor material and methods and in general in reporting, and I will talk about this a little bit later, but obviously poor practice, right? Doing the things in a way that is not adequate to reproduce this data. And obviously we have an ethical problem, right? There's also this fabrication or falsification of research that has been also appearing in the last years. And I don't think this is anything new, but there's some study showing that 2% of the scientists have admitted to fabricate or falsify or modify data results at least once. And there was a huge study and they checked 7,000 scientists. And from these 7,000 scientists, they basically 8% accepted that they have falsified or fabricate data. And I'm pretty sure, you know, this number is even higher because, you know, people don't like to say that they are doing things wrong. So, okay, there is many issues, obviously, but obviously one of the problems is the poor practice in the laboratories, to don't have the proper controls, to don't have the proper reporting. And I'm going to try to explain a little bit about this. So first, I want to talk about something really simple, right? So if you see this video here, if it works, okay, this is a human heart, and you see it's quite complex structure. And then if we try to see one of the models for heart that now people are using, we have iPS-derived cardiomyocytes, right? And I'm pretty sure at least 199% of the people here attending this webinar can see the differences, right? So obviously, we have to take into consideration that we are using models. Actually, we are not using the real organ. And with the models, it comes some limitations, right? Now, because we have in cell culture the tissues, we have possibility of contaminations, and that is an important problem. We can also have growth factor condition that might favor a cell type and sense of the other in case when you have multicellular models. There is a lot of oxygen. Most of the time when we add the media, the oxygen is consumed and we have very few oxygen left for a few days. There's a lack of metabolism and defense. We don't have well, if you have a model for cardiomyocytes, you probably don't have the liver compartment, you don't have immune system and this type of thing. So we have to be we have to understand that we have limiting conditions, and these hundreds of variants we need to be controlled to make sure that our model works correctly for the function that we want to use it, right? So we have to use a model for a purpose, right? But we have to make sure everything works fine. Because if we have a mistake, we do something wrong, there's an important reason. I want to show you why it's important to have a good cell culture practice. Obviously, the most important one is you have the research generated erroneous data, so data that is not right. And that it comes a little bit with the reproducibility crisis, right? Reproducibility crisis. You might have to withdraw the publication. Sometimes if you have some data and then you realize that was wrong, you have to withdraw the publication, and that I will show you later that has important impact in your reputation. Also, you can fail patents, you can waste resources, as I mentioned, with all the money that you lose. There is also the problem that you can have some harmful effect in the employees, because you can get some infections, virus infections into that if you don't take the proper care. And also, you know, the host institutions can go into legal problems. And yeah, and this is always it has two parts, right? One part is the quality of the science, but other part is also the money that the taxpayers are investing, right? And there was this a study also from NIH from retracted papers from 1992 to 2012, where they found that with all these retracted manuscripts, basically they lost 58 millions of dollars, right? So it is important that we actually do a good work, but also if we think into terms of research, you don't want to get into these problems, because, for example, it has been shown that research experience a 91.8% decrease of publication output and large declines in funding after being censored by the Office of Research Integrity. And also there are many cases where people have lost their jobs because they were not doing a good job in the lab, right? And most recently, I just want to give an example that I think is kind of crazy. Probably you guys know about Alzheimer and beta amyloid oligomers. And recently has been a scandal because there was a publication really famous where, you know, talking about these beta amyloid oligomers and the link with Alzheimer. And it has been the main, let's say, the main feature in Alzheimer's disease for the last 16 years. And now there's some evidence indicating that this might be falsified data, because many authors tried to reproduce the data and they couldn't. So I want you to imagine, like, for a main field like Alzheimer's, that has been many research focused on this specific thing. And because it was a mistake or maybe in purpose, we don't know for sure, but you can imagine how much money has been lost and how many people have lost time and resources on this because it was based in something that was not true. So, okay, we talk about ethics and why it's important to have good practice, but let's talk about what we can do to improve and what are the real problems that we have in our lab and how we can solve. And obviously one that is really famous is the microbial infection. And I'm pretty sure if we have some PhD students or some postdocs here, at this point, they are experts in recognized bacterial infection. And I'm pretty sure they open the, the incubator and it's like they smell that food and say, okay, I can smell Bacillus gram-negative 2 days in culture, right? So I think bacteria is kind of easy to identify, still a big problem, and we have to obviously take care to avoid that. But I want to talk about something that is more scary, like mycoplasma. So mycoplasma is amoeboid. It's a type of bacteria that doesn't have membranes. And these guys, the most dangerous thing is they can be in our cultures and we cannot even notice. They don't smell. They are really difficult to see by eye. And there have been many articles showing that these cells, this infection that we have in our cell cultures can change genetic instability, produce transformation, change in physiological functions, and virus susceptibility. So basically, changing the condition in our cell cultures. I think the guys look something like this. I think I took this video from YouTube, and personally, I have never seen them in by eye because, like I said, it's really hard to see them by eye. But they should look something like this, I guess. And it has been really difficult to find them. And I know a few groups that has issues with mycoplasma, and they has to withdraw a manuscript that was already accepted because they realized that in the culture they found this mycoplasma. So yeah, there were some studies, and I like to give examples always. And there was a study where they study different lines of fibroblasts and keratinocytes, and they found that 32% of these lines that they were studying, they were contaminated. And if you see the number, around 70% were contaminated by mycoplasma, 9% fungus, and 6% bacteria. And you would say, okay, this is a long time ago, right? It's not happening anymore. Okay, let's see if we can find the same pattern. There was another study in 2005. They also studied 21 lines from different labs, and they found, again, around 39%, quite similar. They found mycoplasma around 90%, similar to what they found. And also they found fungi 8%, also similar, and bacteria 4%. So quite similar, actually, quite a long time, right? But I'm gonna give you even more information. This was a screening for FDA, and they study 3 decades of cell lines they have, and they found, they study more than 20,000 cells. And they found that 50% of these lines, again, really close to the other ones, right? They found that they were contaminated by mycoplasma. There's also another study where they study a total of 301 cell cultures between 15 laboratories. And they found again that they were highly contaminated by mycoplasma. There is estimation that was published in 2013, where they estimate that 35% of the cell culture currently using in the lab are actually infected by mycoplasma, fungus, or bacteria. So, and the most common way to get this mycoplasma is when you receive a vial from other laboratory, or you buy a line, or maybe from some animal products like fetal serum, things like that. But also it can come from the, obviously from human handling, right? And what is really interesting of this is even in the liquid nitrogen, I don't know how this guy do, but if you put a vial that is contaminated with mycoplasma in the liquid nitrogen, it has been shown that also can transmit through the liquid nitrogen. So mycoplasma is a really serious issue. And I hope you guys are now convinced, and I hope you guys check this routinely, but we will check later because we are going to ask this question later. Another big problem that has been increasing in the last years has been misidentification, right? And this means what type of cells are we using when we are in the lab? And this is something that has been a problem in the past, but it has been showing an increase over the time. And this is probably not because we are worse than before. It's because we have better systems to detect when we have misidentification. So now we can use short-handed repeat DNA profiling, and then we can identify quite easily what type of lines do we have. So And this has been a problem, and I want to show you an example. This is an research that was done in China in 2017. Basically, they studied 278 lines from different laboratories, and there were tumor cell lines, and they found that 46% of these lines were cross-contaminated, meaning that they have other cells a part of the cells that you want to have, and also misidentified. So maybe they have other cells that actually they were not the ones that they were expecting. But the most funny thing for me is they found that 67% of these lines actually were contaminated with ELA cells, what I found amazing. So basically, these cells are so easy to reproduce and so resistant that if you have one cell going into one vial, into one plate, it will, over the years or over the months, it will take over the other line, and basically in the end, you will have these ELA cells, right? And I'm pretty sure you guys are thinking, well, this was in China, you know, here in Europe, we have better controls, blah, blah, blah. But I want to mention that I know a case, I could not say the names, in a really good university that they got a line thinking that they were a brain line, right? And they were working in this brain line for 3 years. And after 3 years, they do the identification, and they found out that actually this line was from liver. So these things are still happening in our regular work, and also in Europe. So be careful. And if you get a line from a lab now, especially with iPSCs, where we have labs that have like 20 lines or 200 lines, and it's really hard to keep up which line is what. And especially with IPS, where all the morphology will be the same, right? So you might want to get one line that has a mutation, and in fact, you are getting another different line. So I will suggest you, every time you get a line into your lab, you identify this line. And now I think we are gonna have a poll. So the questions are, how often are mycoplasma tests performed in your laboratory if you are working in a laboratory? And the second question is, how often are these your cells, you know, are checked by integrity or using STR to identify the cells that you have in your lab? Okay, so good. It's not too bad. I had worse, but yeah, it's still like I'm pretty sure some labs, at least 10% of the people here don't do this often enough, and that is a problem, right? So we have to be careful. In cell identification, I saw even higher. So this is something that you have to take into consideration in the future. Okay, so other problems I want to mention is related with genomic instability. And obviously, when we work with some tumoral lines, we got something. This is from ELA cells. As you can see, it's kind of crazy, right? How many chromosomes have for different aspects. So, you know, obviously with the tumor lines, we always going to have this problem. But now also it has been shown that genetic instability in pluripotent stem cells has been detected since 2000 when you do the karyotyping after, you know, the derivation of the expansion of these cells. And also when we are doing, for example, reprogramming of somatic cells into iPSCs, we can found different genetic problems like chromosomal aberration, aneuploidies, and deletion of different parts. So if you are doing also iPSCs, it's not that you are gonna remove this problem of genetic instability, right? So this is something important also to measure when we reprogram some cells, or when we make some stem cells, or even when we keep them for a really long time for different passages. And then I want to talk a little bit about reporting, and I want to give this case. This is a work that has been done by Tracy Weisgerberg from the Berlin Institute of Health. And I found this really interesting because there was this case where, you know, they are working a lot on trying to see how people report the materials and methods. And I found this quite hilarious that, for example, this is a case from Daniel Gonzalez, where he posted that in 2017, he read a paper, and then in the paper described the device were fabricated as previously described in reference 8, right? So then he got to reference 8, 2015, and it says basically the same, but reference 4, he got to reference 4, the paper is from 2013, again the same thing, reference 2, and then when he arrived to the real or the prime manuscript, then it says the bytes were fabricated with conventional methods. And I don't know how long, guys, you've been in science, but I think probably you guys have encountered this issue. And the worst thing is we face this problem, we complain, but then when we have to do it, sometimes we also put, like as it was described in this paper on so on, right? So I think we are now facing also this problem and I think this is quite linked to the reproducibility of the data. So we have to make sure that we have a good reporting. In the GCCP guideline, they have some indications how to improve this reporting. And I'm also aware that there's other activities. For example, the European Commission now, led by Sophia Leite, they are trying to develop methods protocol initiative where they have people from the publisher, funders, and scientists, and they are trying to figure out how we can implement better methods and requirements for reporting in the manuscripts. So if you are interested in something that, be aware that something is coming from the European Commission on this sense. Okay, so I hope I have convinced you that some of the things are happening in the labs and why it's important to have a good cell culture practice. And as I mentioned before, this is guidelines to tell you, a little bit, some advices on how to do a better work. And a little bit of a story of GCCP. This started in 1988, so a few years ago. And we're starting talking about this, that we need to find a solution for good cell culture practice. And then there were two parallel initiatives in Germany in 1996 and 1999. And slowly it moved into, declaration toward good cell culture practice by Gerhold and Thomas in 1999. And then in the end, it derived in these two reports that were published in 2002 and 2005. And this is one of the manuscripts that I mentioned. But because things have been changing in the last years, right? We have now IPS, and in 2005 they were not present in our cell cultures. We have now 3D models. We have on-a-chip technologies. They have these new technologies are coming with obviously new challenges. For example, how to maintain the iPSCs, how to control the genetic instability, the different reprogramming methods, different protocols, how mature is our model, if is our model very complex, how we do the quality control of this model, right? So that's why I think 2015, we started with the GCCP initiative, where we have partners from different institutions, including European Commission, NIH, and some private companies, EPA. And the idea was to generate an update of the GCCP guideline from 2005, also create some scientific discussion to see, okay, what is important, what we need to address, and promote activities from GCCP, and also to educate. And I hope that is something that I'm trying to do today. And this was this second step was initiated at CAT, Center for Alternatives to Animal Testing, and now is moving into evidence-based toxicology, trying to be independent. So this has changed since April 2018. So in the first phase, we have different workshops where we talk about what are the new technologies, how we can implement these technologies. And then after that, we spend several years, and it was tough because we had to collect all the information, put all together to generate these new guidelines that now is published. And I really recommend, if you're working in cell culture, that to have a read, because it gives you really good information that you can adapt into your laboratories. For example, I just want to give some example. Here we see a table that is included in the document that talk about key elements to ensure the successful cryopreservation. So basically here you can find what are the different things you have to control, what are the protocols, how should be the protocol, what is the storage conditions, and different things you can control to make sure that when you do this cryopreservation, you are doing it in a proper way. The same way we can find other tables. For example, as I mentioned, one of the principles is related with safety. So it can give you different examples of how to can preserve the safety and the handling of the cell tissue and also the operator, right? And as I mentioned, we have tried to incorporate also the new technologies. So we have also some tables related with iPSCs or stem cells. What are the different markers that people are used to make sure that you have pluripotency and self-renewal genes? what are the controls you have to do to make sure that you don't have these genetic instability. So a series of recommendations that will be really useful for the use of iPSCs. And we have also some text related with complex cultures like organoids, human chip technologies, and so on. So basically, it gives, I think it gives a general information about what are the things to take into consideration to have a good cell culture practice in your laboratory. If it's not enough, I want to mention that it's even a more complicated guidance. This is a guidance document on good in vitro method practice, GVIMP, and this was generated by European Commission and endorsed by OECD. And this is a really much complex guidance on cell culture practice, but this is, I guess, is more into more like into GLP type of laboratories that and GCCP is more for into academia context. There's other activities coming, and I'm gonna be finishing soon. There's other activities coming. There's one recent publication of quality criteria for in vitro human pluripotent stem cells, where it gave a good advice of quality controls that you can use. As this is part of the principle 2 in the GCCP guidelines for stem cells. And now we are trying to generate one for quality management plan for microphysiological systems, including organoids and microphysiological systems. There has been also some initiatives, for example, with Thomas Hartung, Marcelice, and so on, related to good in vitro reporting standards. And as I mentioned, the European Commission is also doing some activities to try to promote these methods and protocols. The steering committee is formed by me. That was the former coordinator, but now it has passed to Rob. And we have different people that we have Sandra that did that was the main author for the first guidance, but we have really important people that has been from the beginning, like Gerhard, Thomas Hartung. Glenn Stacy has been really important on the work that we have produced. And obviously also marginalized in the update of this GCCP. But we have an extensive scientific committee, so this is not the only authors. We have many others that has been contributed over the years. So as a conclusion, I think it is clear that we need good practice in the laboratory if we don't mess up and get some issues. I think GCCP can provide good guidance for good cell culture practice, and I recommend everyone to have a look on these guidelines. And I think it is clear that not using good practice can derive in several problems that can be ethical, legal, or economical, as I showed you in the beginning of the presentation. And with that, I did 30 minutes. I want to thank you, everyone. And if you have questions, you can send me an email or you can ask now in the question Q&A.
Thank you, David. And meanwhile, also Marcella is joining me.
So perfect. He can answer all the questions.
So please, and meanwhile that you think about your question, and you can use the Q&A function. There is also a last poll that we would like to pose to you. And it's indeed something that was mentioned during the talk. And I also got this question, where you think that the GCCP should be implemented, at which level, who should support this?
Okay.
So quite many, they think it should be at the level of individual organizations.
Mm-hmm. Okay, so I'm just gonna give you my opinion. Obviously, you know, as a scientist, you want to do the best, and you should do taking care of yourself, obviously, on that sense, right? But I think also journals and funding bodies could help with this. And I think there's some now initiatives. We have River and Arrive that are minimum standard reporting for in vitro and for animal studies. And I hope this will be what now, for example, the European Commission is trying to see is how journals and funded bodies can use this as a kind of requirement for publications. And this is hard for the journals, right? Because if you put too many requirements, then people don't want to publish in your journal, right? So they have to be, you know, a consensus between all the main publishers to do that. But I think that will help also, you know, because as most of the time scientists are overloaded, I kind of feel that if we are not obliged to do something, sometimes we don't do it, right? And obviously that depends on the ethics of each person, but I think definitely we need some push from some of the authorities or publisher funding, so some kind of push. I don't know what you think, Marcel, but
just briefly, so if we compare it to the animal experiments, there has also been the situation before the Arrive guidelines, and it was found that the reporting and the practices are at least not good enough documented, sometimes also not good enough. And nowadays, most journals publishing animal experiments require that you go through a tick list there. So it wouldn't be so different from this.
Yeah, yes.
David, we have one question from the participants. I will read it. So most of the cell lines go through a process of selection against antibiotics, and so they have some sort of resistance. So at the cellular level, wouldn't it be a kind of immunity for their survival that could be correlated to immunity in the in situ conditions? So do you think mild contamination would not make any difference? So like having the mycoplasma is the normal situation of cells, so maybe we should leave it. I don't know if this was the.
Well, I think, well, I'm not sure if I understand 100% the question, but I think you want to control what you have, right? And the main problem of mycoplasma is you don't know that you have it, right? And it has been many research showing that mycoplasma actually alter results, right? So I will think that no, I will think it's better not to have it. That's my personal opinion. But yes, it's true that antibiotics are also an issue because there's some indication that also can, select some specific populations or help some in the differentiation, for example, things like that. So if possible, we should try to culture the cells with antibiotics, right? But it's not always so easy.
Yeah, indeed, the antibiotics topic is quite important and maybe underestimated. It is also mentioned in the GCCP. How to handle this?
Yes, it is also in some advices. Yes, I mean it's like if you read the guideline, it's try to give advices more like force people to do a specific stuff, right? So it's more like try to you should do that. This is maybe a recommendation for improve the quality conditions of the good practice in your laboratory.
Yeah.
Last century technology. It's like people used to have antibiotics, last century. So I haven't heard of a cell line which would be a problem to be cultured without antibiotics. This is when cells used a lot of primary cells. Then, of course, during the isolation, you can get something in from the fur of the animal or whatever. But on stem cells, it's normal to be without antibiotics, and on most cell lines as well. So there was this question on the immunity. Of course, you cannot compare immunity on cells. So you would call that resilience. That is a changed state that reacts differently to a stress, and that is very fundamentally different from immunity, where you have specified cells that recognize something and that helps the overall body. Just to clarify things.
I think we have one question from the Q&A from Jana Kuhn, which I think is very interesting. And the question is, if you can at least be sure when you purchase a cell line from a supplier that it is the correct cell line, or should you also check these cell lines right away after the delivery?
Well, I can only give my opinion. In my opinion, because we have had some conversation with Glenn and Stacy that work in the UK stem cell bank for a long time. They also found this problem of misidentification sometimes, and obviously anyone can make an error. And when you get a line, you normally want to do like a stock bank with this. It's called token bank with this cell. So. And also now it's not so difficult to identify the cells, right? So what I will recommend if you have a line purchase, I will definitely do the identification. Probably I will expand and do the identification and make sure also check for mycoplasma and make sure that all my token bank is perfect in case I want to use it in the future. But that's my recommendation. Obviously, stem cell has high control, so it's not the same receive them from a cell bank that receive them from another lab that you don't know the conditions. But with the cost that is now not so high, I will recommend to do it. But that's only my personal opinion.
Oh, there is well, let's maybe take for the second one. That is, what will be your recommendation for the frequency of mycoplasma testing?
For example, in our I can only talk about our lab. In our lab, what we do is every time because we have a stock bank, right, where we have these cells that we are supposed to have under control. When we put a vial out, because we normally expand the cells after we do our cultures, because we use neuroprogenitor cells to differentiate into brain cells. So every time we expand these cells, we check for mycoplasma. So let's say we do this every 2 months. We test every 2 months. If we do every 3 weeks, we test every 3 weeks. Like I say, now it's getting quite cheap and it's not super expensive. So, and for my sanity, I prefer to do it quite often. But I think if you, it depends how many lines you have and so on. But I think maybe once every 3 months will be something. It depends how much work you want to lose, right? You think if I do every year, but then I found mycoplasma all this year is, to the bin. So, it depends how much you are willing to throw away in case you get mycoplasma, right? So in our case, because they are quite expensive, we try to do maybe at least every three months.
Perhaps one small point. It depends, of course, also on the exchange of your lab with other labs. If other cells are coming in, if other reagents are coming in, there's a higher risk level. Especially if you receive cells from outside, it's usually a procedure to put them in a quarantine incubator separate from all the cells. And definitely every time to do the mycoplasma testing if something comes from the outside, and then only put them into your normal routine.
Well, let's keep the mycoplasma topic. There is a question if is mycoplasma contamination only an issue for human mammalian cell culture, or would mycoplasma also infect avian or reptilian cells?
Well, actually, I'm not sure. I'm assuming it will be also mycoplasma for this type of cells. But Marcel, do you know better this? I don't know. I guess so. I have never worked with these cells, so I'm a bit lost. I don't
know. Okay, then let's go back to one of the first. It's a bit about more the last topic. It's well, congratulations to your work, and unfortunately, I don't see GCCP being a priority in terms of funding, at least in Spain. Any ideas on what to do to promote it? We have discussed it a bit, but yeah.
I think. I mean, it's not. It should not be related with funding, right? It's supposed to be related to how good you want to do your things in your laboratory, right? So I think to implement GCCP doesn't have to be expensive, right? So I think using the minimum, it obviously depends on the money your laboratory has, right? But like I say, now things like mycoplasma testing are getting cheaper, and putting quality controls every time you do something, it doesn't mean that you have to do extra experiments. But for example, if you are cultivating some cells into neurons, you have to make sure that every time you make neurons, so you can, for example, check the morphology, or maybe have some small assay before you perform your experiment to make sure that they are electroactive or something like that. So establish some quality controls that you are sure that what you are performing and the things that you are getting are in the good quality. And I think in the end, this is going to save you money a long term, because if you have some problems, then you will waste a lot of time and money. And time is also really expensive in science. So I don't think it has to be related with the quantity of money. It's more about, establish good protocols into your laboratory. This is something you don't need to spend money. It's just put a little bit of effort to make sure everything works.
But maybe I think the question also was a little bit in the other direction. and it goes back to the last poll. So should the funding bodies make it obligatory that you follow GCCP for when they give you money?
Yes.
And I think in the poll we asked, should it be the journals or should it be the funding body? It could also be both. So funding bodies could only basically accept publication in a journal that makes it obligatory.
From my point of view, it's hard to it's hard to control good cell culture practice, right? Because what is exactly, so you can control it, reporting, right? I can say I want this to be reported to make sure that you did a good work. That's something you can do. So we can put the standards in reporting, right? And that should be something that should be pushed by funding bodies, publications, and so on. But good cell culture practice, it's many things. So how am I gonna make sure that someone is using the lab code in the lab when they are doing, cell cultures? So I think that's more difficult to control in that term, right? You can say in your publication, yes, I do good cell culture practice, but it doesn't mean that you are doing right. But in the reporting, this is something we can implement. We can ask for mycoplasma tests. We can ask for identification. We can ask for methods. These type of things is what I think should be pushed by funding bodies and by also publishers, right? In terms of getting good quality in their journals.
Full confirmation. It's not a method. It's a kind of a culture. It's exactly as you said, David. It's doing good science, and good science is all these principles. That's why they are not rules, they are not laws, but understand your system, know what you're doing, use the quality that's required to produce data other people can rely on. This is why you do it. And then there is some peaks of the iceberg or something that you could perhaps check in a journal, like cell identification and mycoplasma testing, but it's a general culture, and it will just make science better.
Yeah, there has been also some, comments to see how to I don't know how to implement this in terms of how to say it, in terms for publications, right? Like, for example, there were this idea of get a certificate, right? So someone go to audit your lab and say, okay, you have good cell culture practice, and then you kind of prove that GCCP is using in your lab. But I don't think it's work for academia setting, because first, no one is going to pay someone to come and audit and have the money to do that, and also it doesn't tell you that you are going to keep this for the rest of your years working on that, right? But I know, for example, for GCCP, now there's an initiative where they have kind of like a branding where they audit your lab and they give you, like, implement this GCCP into your laboratories. But obviously this is for more GLP laboratories that, control laboratories that they have the money to invest on that, and they want to prove that they have quality control, right? But for academia, it's kind of hard to try to do that, I think.
Question in the Q&A: what are the absolute necessary measures that should be considered for the cell identity testing?
Well, I will use STDR, but I guess also, maybe sometimes with karyotyping and these type of things, there are different variations now how you can identify your cells. Maybe you don't need a super high level, right? But it's not so expensive to go into like very detailed identification nowadays, right? Maybe it costs $300, 300 euros. So yeah, it depends how deep you want to go. Like sometimes I know some labs do karyotyping because maybe they have the technology in the lab and they can see, for example, if you have a trisomy and you want to check that it's kind of easy, right? But if you got to something more deeper, like mutations, things like that, you have to go into more detail. So it depends a little bit what you are interested in.
Maybe in the meantime, I can ask a question.
Yeah.
As you and also Marcel said, it's mostly about culture. So where should we start teaching GCCP in the educational, and is this already implemented in university programs?
I think it should be before people enter into the lab, right? So people should be trained before going to the lab, for like we do for different techniques. They should also be trained in ethical issues, but also in GCCP and these type of things. So people, when they enter into the lab, they are ready for do a good job. So for me, that's the level we should do. And Unfortunately, I don't know much. I don't know any case of, implementation of GCCP. Obviously, separate, yes, right? There are some institutions where they have a program for, talking about safety, or there's now an initiative trying to do also for reporting SOPs and stuff like that. But I think as a package, GCCP, I'm not sure if there's any. Maybe maybe Constant University, but I don't think I have seen any case at the moment on this. We are trying to do some talks, try to promote this, but obviously we are not so many. So, but I think it would be cool to have like more implemented in the like education, like the same way that PhD student might have a course in SOPs, maybe in GCCP also.
I think it's just heterogeneous. So there are some universities, some study courses where there's a good cell culture course, which also teaches all the principles, sometimes under different names and different headings, but these principles are very fundamental and general. Sometimes it's the culture of big labs that when students come in, they get some initial training, or they get training from colleagues, and they get these principles, and in other places it's not existing. So probably the focus should be to make sure that everybody gets at least a minimum kind of level of this. In good places, it's often in place, or in some master studies or bachelor studies, even it's in place.
Okay, I think this is also a good way to close the webinar. Think about next generation and trainings. And so I would like to thank again David and Peter, Marcel and Silke, and all of the participants for all the inputs, and also take the chance to share the next appointment for the 3R webinar. So we will have in October a talk about small refinements to improve laboratory animal welfare by Pauline Kirchhoff. And in November, we will have a panel discussion around implementation, new approach methodologies on NAMs in industry. So we will have 4 different speakers from industry, and we will have a chat with them to check at which point we are on NAMs implementation. So I will, with this, I will like to say goodbye to all of you, and I hope that you will join again in the next webinars.
Thank you. Thank you very much.
Thank
Have
you,
a good evening.
Bye-bye.